Hi everyone, I'm here with Dr. Rob El-Maraghi from Barrie, Ontario. We're at ESMO 2015 in Vienna, Austria.
Yeah, hi. Thanks, Charlotte. I'm Rob El-Maraghi. I'm a medical oncologist at the Simcoe Muskoka Regional Cancer Centre at the Royal Victoria Regional Health Centre in Barrie, Ontario. And we are here in Vienna, Austria. It's the European Cancer Congress 2015.
What is the standard treatment regimen given to patients with stage 3B or 4 squamous cell carcinoma of the lung after progression on platinum doublet chemotherapy?
Well, at present, it really depends a lot on performance status. For someone who has got a good performance status and is still eligible for second-line therapy, typically most clinicians would offer a single-agent chemotherapy such as docetaxel. For those patients, however, who are poorer performance status, an EGFR and TKI might be a consideration.
Thank you. In LUXLung 8, the median overall survival was significantly higher for patients taking afatinib compared with erlotinib. Can you comment on this result?
I think it's quite interesting. Again, an EGFR TKI is certainly an option for a patient. When you have different options available, it's really nice to see if there are any significant differences. And of course, the things we're looking for are whether or not there's any differences in, say, quality of life or in some of the harder outcomes, such as overall survival. So to see some slight differences between the agents will help us to tease out whether or not one agent versus another might be more appropriate for the particular patient we're wanting to use it in.
Will the overall survival data change how you treat patients with squamous cell carcinoma of the lung?
I think the information that we have is compelling. But again, it will be often a decision-making process between the particular agent and the particular patient that you're trying to apply that agent to. So while the overall survival information is quite important, it will have to be taken into the broader context of the rest of those parameters.
Quality of life and lung cancer-related symptoms were found to be significantly improved in patients receiving afatinib compared to erlotinib. Cough, dyspnea, and time to deterioration was significantly prolonged. Can you comment on this?
I believe that quality of life, particularly in a situation where you have a patient who is not curable, is a very important endpoint. Anytime that we can help people to feel better, my position is that they often live longer and they will often have a much better outcome because of the fact that they are out with their families, they're out with their friends, and they are not experiencing all of the side effects that we often think about when we think about someone who's got a very advanced lung cancer. So these endpoints for me are particularly important and also quite compelling for the use of one agent versus another.
Thank you. Overall, the adverse event profiles were comparable between the two drugs. 57% of patients in either group experienced grade 3 or higher adverse events. However, patients taking afatinib had a higher incidence of grade 3 diarrhea and grade 3 stomatitis, whereas patients taking erlotinib had a higher incidence of grade 3 rash. Based on these results, can you comment on whether patient quality of life is different between these two drugs?
I believe that, again, that there will be certain times when you may wish to use one drug versus another based on the perceived side effect profile. I think the most important aspect here, though, is to remember that these are very manageable side effects. With proper patient education and some of the tools that we have available to us, and perhaps being a bit more proactive with the use of these tools, such as the appropriate hydrocortisone and clindamycin creams, or the use of corticosteroids, or the use of antidiarrheal agents, for instance. I think we can definitely approach these patients in a very proactive manner and help them with their side effect profile, whatever that may be. Another thing to consider is obviously that if there is a particular problem with relatively significant side effects, that dose interruption and even dose reduction is quite a very appropriate step to take in some cases.
What is the take-home message from the LUXLung 8 trial for physicians?
I think the data suggests, at least in the context of the LUXLung 8 trial, that when you directly compare the two agents, in this case afatinib and erlotinib, in this particular group of patients, this trial suggested that there was both an improved outcome for quality of life as well as for the parameter of overall survival.
What does this data tell you about the ERB family of blockers versus first-generation TKIs?
Well, what this data suggests is that the family blocker is a superior agent to the first generation TKI in the context of squamous cell carcinoma of the lung.
When you look at data from a randomized clinical trial, which outcomes are important to you?
For me, it depends on the trial we're looking at, but in the setting of an advanced lung cancer, the parameters that I'm most interested in are quality of life, progression-free survival, and overall survival.
At ASCO 2015, a meta-analysis of 6 random clinical trials of gefitinib and erlotinib by Lee and colleagues demonstrated that despite significant PFS benefit, there is no OS difference with first-line gefitinib or erlotinib when compared with chemotherapy in advanced non-small cell lung cancer with common EGFR mutations. Can you comment on that?
Well, to my understanding at least, one of the reasons why we didn't see an overall survival advantage in many of the earlier trials was because of the fact that crossover design was allowed. And in some other cases, there was the ability for patients to have second, third, and sometimes even fourth or longer lines of therapy. And the feeling was that the EGFR driver mutation was so important that if you were to have these agents in a later line of therapy, that the benefits would still be obtained. Of course, there are a number of different ways to sort of tease out this information and try to decide when you might wish to use them in your own treatment strategy. But of course, you have to take into consideration whether or not patients will actually get onto later lines of therapy if they're not used in the first line. And of course, it is very important that patients who do have an EGFR mutation are at some point treated with one of these agents.
Here at European Cancer Congress 2015, a retrospective analysis by Choi and colleagues demonstrated that there was significantly better outcome of gefitinib-treated non-small cell lung cancer patients with deletion 19 compared to those with L858R. Patients with deletion 19 had significantly longer 2-year overall survival rate compared to those with L858R mutation. Can you comment on this?
Yes, this is a really interesting development that we've discovered over the last year or two. And that is that there is a difference in the response rates and outcomes for patients with different mutations. It is not a one-size-fits-all. It is actually some sort of a distinction between these two that are leading to differences in the manner in which the agents are actually affecting the TKI binding pocket and therefore the overall outcome of the patient. And it does seem that there is a difference in the sense that the patients with deletion 19 mutations tend to respond better to EGFR TKIs and overall do better with prolongation of survival.
What are the advantages of head-to-head trials?
Head-to-head trials are quite important because they do help us to distinguish differences in outcomes, obviously, number one, as well as to help to tease out when you may wish to use one agent versus another based on a side effect profile that you can actually directly compare in the same patient population.
The CTONG 0901 head-to-head trial presented at World Lung 2015 found that erlotinib was not significantly superior to gefitinib in advanced non-small cell lung cancer with either deletion 19 or L858R mutations in response. PFS and OS had similar toxicities. What are your thoughts on this?
I think it's quite interesting. It does go to show that the earlier TKIs that we've had access to, including erlotinib and gefitinib, are fairly similar, and that you can make treatment decisions based on agents in terms of their side effect profile and their quality of life profile if you wish to do so. It's interesting also how regardless of whether or not there is a deletion 19 mutation or a 0.21 mutation, the outcomes are very similar, and again leads you to think about these other parameters when you're making treatment selection.
There was no significant difference in median PFS. Could you comment on this?
Yeah, sure. So basically what this is telling us is that when we use either an EGFR TKI such as erlotinib or gefitinib, that their outcomes were similar in terms of their PFS. Like I've said throughout this interview, it does go to show that perhaps the earlier TKIs that we've had access to may be better selected based on their their overall patient tolerability profiles rather than their ability to perhaps impact one deletion or shall I say one mutation or the other mutation in a greater or lesser fashion.
Earlier this year, Yang and colleagues published in Lancet Oncology an analysis of data from LUX Lung 3 and 6 trials, which were conducted in patients with EGFR mutation-positive advanced lung non-small cell lung cancer. They showed that OS was significantly longer for deletion 19 patients taking afatinib versus those receiving chemotherapy. Patients taking afatinib in LUX Lung 3 and 6 also had tolerable and manageable safety profile. Now the results of LUX Lung 7, a randomized open-label phase 2 trial of afatinib versus gefitinib as first-line treatment for patients with advanced EGFR mutation-positive non-small cell lung cancer, will be presented at ESMO Asia in December. Based on your experience and previously published data, what do you expect the results to be?
Now this is going to be a very interesting question. What we know from LUX Lung 3 and 6 is that if you combine the data, that there seems to be a significant difference for those patients who harbor a deletion 19 mutation. And for the very first time, we actually saw an overall survival difference with the use of an EGFR TKI, in this case afatinib, compared to the current standard chemotherapy regimens. We also know from previously published data that drugs such as gefitinib or erlotinib have thus far not shown a difference in outcome when compared at least head-to-head when you use these for either a deletion 19 mutation or for a 21-point mutation. So if we were to hypothesize, it would be very interesting to see if that difference still holds. And that being that the drugs, the newer generation drugs such as afatinib, whether they still have an improved outcome compared to earlier drugs such as gefitinib in terms of the deletion 19 patients. I guess at this point it's pure conjecture, it's hypothesis generating, and we'll need the trial to see if that actually pans out.
Are you looking forward to seeing this data?
Yeah, as I just mentioned, I think it's going to be quite important to see. There are some clinicians who will actually start to make decisions based upon which mutation we are seeing. Of course, we do have to bear this in mind with the side effect profile of the drug, comfort level of the drug, and the particular patient you're using it in to ensure that all of these parameters together lead to a better outcome. But overall, I would say that yes, I'm certainly interested to see how this pans out.
The findings from the IMPRESS trial were presented at World Lung 2015. Investigators found that patients who were T790 mutation positive, median PFS was shorter for gefitinib and chemotherapy compared to chemotherapy alone. In patients who were T790 mutation negative, PFS was longer with gefitinib and chemotherapy compared to chemotherapy alone. What do you think about that data?
Well, it certainly goes to show that when we, as we are learning more and more about the biology of these agents, how they work in our patients, that there are important things that can occur, specifically with the development of newer mutations, such as in this case T790M, which typically leads to resistance of some of the first-generation TKIs. So when you have outcomes like this, it does certainly help us to begin to understand when we may wish to use one type of therapy versus another, and leads to the importance of the development of new clinical trials, which are looking at agents that can specifically affect some of these mutations that are now coming to light.